So just to review what we were going over at the end of the last video, we were talking about how Mendel did his experiments. So you've got true breeding purple flower crossed with a true breeding white flower that gives you all purple flowers. And then if we look at the next generation, so that all purple flowers were going to be the F1 generation, then the next generation, called the F2 generation, would be if you allow these purple ones to self-pollinate, and you'll have that 3-to-1 ratio. So all of this information was going to be stuff that Mendel was going to use to determine how genetics work. And if we look at this slide right here, you can see this is showing that 3-to-1 ratio. Now these words over here we're going to go over in just a second, but I just wanted you to see that. Okay, so let's go through the model of heredity that he came up with as a result of what he found with those flowers. First thing he found was that parents do not transmit their physiological traits directly to their offspring. And what that means is you are not going to get everything expressly from your mother or everything expressly from your father. You're going to get a mixture of things. For each character, which is a general category like eye color or hair color, an organism is going to get 2 alleles, one from each parent. So that means you would get an eye color allele from your dad and an eye color allele from your mom. And then these alleles are going to account for variations in inherited characters. So there's a couple of different ways that things can be set up as far as the alleles go. If they're going to have 2 of the same type of alleles, we call those homozygous. Zygous is talking about the alleles. Homo means same. If they have different alleles, then they're called heterozygous. Hetero means different. Zygous is talking about the genes. So another thing that's important in his model is that those 2 alleles for each character are going to segregate when gametes are produced. So this is going back to that meiosis idea, and all this is just saying is that the 2 alleles don't stick together. They're going to go into their own gamete, because remember the gametes have a haploid number of chromosomes. And then a little further down, the last little part of his model, if you have 2 different alleles together, that means you would have a dominant and a recessive one. The dominant one is going to be fully expressed. The recessive allele won't even show up at all because it's getting covered up by the dominant one. Now, we're going to be talking about the genes that something has and also what they look like physically. So if we're talking about the genes that an organism has, that's called the genotype, and that would actually be what alleles that organism has. The way that that physically looks is called the phenotype. Okay, so in this example, I've got the genotype being big B, little b, which means the phenotype would be brown eyes because brown is dominant over blue. All right, we're going to review that in just a second. So the way that he interpreted his results, I've touched on a little bit. Dominant alleles are always going to be written in an uppercase letter, so you see a big P here. Recessive alleles are going to be written with a lowercase letter, which is that little p you see here. So if we say that something is homozygous recessive, okay, let's take that apart. Homo, same. Zygous is alleles. Recessive are going to be the lowercase. Then their genotype, which is the genes that they have, are going to be little p, little p. If you have something that's called homozygous dominant, that's going to be 2 of the same that are dominant. So that would be big P, big P. And if something is heterozygous, we're going to say it's big P, little p. Okay? Now, I purposely use the letter P here because it's very easy to mix that up, isn't it, to see the difference between big P and little p. So what I want you to notice is that when you write these things out, use the cursive version of the lowercase one, and that way you won't have a mistake. Now, if it's something like A or R, those uppercase and lowercase letters look so different from one another that that shouldn't be a big deal. All right, so let's look at my PowerPoint here again, and let's talk about how this works. So this is the F2 generation we were talking about. And what he found was that he had 3 purple to every white when that happened. So the white one, since that's recessive, would have to be little p in order for that recessive allele to show up. And then these other ones, we've got big P, which is homozygous, and then we have 2 big P, little p's, which are heterozygous. And we're going to come back to this in just a second. All right, so let's just do a little review. If something has the genotype of big A, what do we call that? Then we also have to think about if we have a genotype of little a, what do we call that? And then finally, if we have one of each, big A and little a, what do we call those? So let's start at the top and go through the vocab that we just learned. So big A is going to be what we call homozygous dominant. Homo meaning same, dominant meaning the capital letters. Little a then is going to also be homozygous, right, because it has two of the same, but it's going to be recessive, right? So it's going to be called homozygous recessive. And then this last one here, you have one of each, so they're different, so those are just simply going to be called heterozygous. So we don't need to say heterozygous dominant because it's just assumed that the dominant is going to cover up the recessive. Okay, so some other terms that we need to talk about are going to be genotype and phenotype that we talked about in the notes. So here you can see there's someone with blue eyes, and then there's someone that has brown eyes. So there's two words that we're going to talk about, phenotype and genotype. So for the phenotype of these, the phenotype of that top left one is going to be blue, and the phenotype of this one is going to be brown. Now the next word we're going to use is the genotype. So what genes are they going to have, or what alleles do they have? So the blue-eyed person, since that's recessive, there's only one combination they can have, and that's going to be little b, little b. Okay? Because for a recessive trait to show up, they both have to be recessive. Now for brown eyes, the genotype could be two different things. You could either have big B, or you could have big B, little b. Just by looking at that person, you can't tell if they have big B, or big B, little b, because that's just, you can't, because that dominant allele is showing that up. All right, let's go back to the notes and talk about a Punnett square. So you're already working on Punnett squares at this point, and Punnett squares are going to be a way that you can show a cross between two individuals and predict the probability of certain types of offspring. So going back to here, if we look at this, no, not that one, this one right here. Let's make this a little bigger. Okay, so we have our true breeding purple and our true breeding white. So we have a homozygous dominant being crossed with a homozygous recessive. So what's going to happen is we're going to cross big P with little p, and we're going to end up with offspring that are all purple. And that's because all of the offspring are going to be big P, little p. Okay? Now, that's that F1 generation. So all of the F1 generation ends up being heterozygous. Now we have that F2 generation, and so what's going to happen there is we're going to have this guy and let it self-fertilize for a generation. So that means you're crossing something that is big P, little p, with something that is big P, little p. So two heterozygous crossing. So the way you do a Punnett square is you just line up the big P and the big P right here, and so you have big P, then you line up whatever is going to be in this box, so big P, little p. Same thing here, big P, little p, and then in this corner you have little p, little p. And so that's how you end up with that 3-to-1 ratio. So this thing right here, oops, is a Punnett square, and I'm actually going to attach another video on content. So there's a little tutorial about how to do Punnett squares of different types. Okay, back to the notes. Let's talk about a couple of laws that Mendel came up with. First one is segregation. So we've already talked about segregation a little bit, but this law of heredity is talking about the fact that alleles are going to be their own separate entities when you're making gametes, and they don't come across as like a package deal. So if someone has blonde hair, it's not necessarily meaning they're going to have blue eyes. So those two don't come together as a package. Now, underneath here I've written something called a test cross. And a test cross is something that you can do if you're unsure about the genotype of an individual. Okay? So right here we've got a purple flower. And we don't know if that purple flower is big P, or if it's big P, little p. So a test cross is a way of testing it to figure it out. So anytime you do that, what you're doing is you're crossing something with a dominant phenotype, but you don't know the genotype. So like someone with brown eyes, you don't know if they're big B or big B, little b. And what you always cross them with in a test cross, always, is with a recessive phenotype. And the reason is because you know the recessive one's genotype, which is little p in this example. So now we're going to do our possibilities, right? So the purple flower could either be big P, or it could be big P. And then we're crossing it with something that is little p and little p, right? So then we can look at the results. If half of them end up showing that recessive phenotype, then we know that the genotype was big P. If they all turn up purple and showing the dominant phenotype, then we know that it was big P. So that right there is a test cross. Okay, the second law of heredity is talking about independent assortment. And once again, this is saying that we're not going to have any sort of packaging of gametes together when gametes are formed, or alleles together when gametes are formed. And underneath here, I've written monohybrids and dihybrids. Remember, hybrids are going to be crossing things over that are opposite phenotypes. And so you can see here, a monohybrid is going to be an individual that's heterozygous just to one gene, so big Y and little y. Then you can see we have dihybrids, and dihybrids are going to be ones that are heterozygous for two genes being studied, so big Y, little y, and big R, little r. So that's why it's called a dihybrid. Now, one thing that's important about inheritance is it's obviously not going to follow all the rules all the time. So dominant isn't always just strictly dominant, and recessive isn't always strictly recessive. So there's a couple of things that can affect Mendelian inheritance, and on the next video, we're going to talk about that.